But to end then, I would like to spend a little bit of time on what I perhaps find as the most exciting sort of future development of this. And that if we can get to sort of things, functional things that are unique to modern humans relative to our closest relatives. So things that have changed here, which we can now identify when we have the high-quality Neanderthal and Denisovan genomes, that have changed here, yet spread to everybody today, no matter where we live on the planet. So things that happened here and became fixed in humans very recently in our history. And why are those sort of changes particularly interesting? Well, I do think that there are some things that have happened when modern humans came that have really set us off on a totally different historic trajectory, of course. So if you look at stone tools, for example, that the Neanderthals made 300,000 years ago, when they appear, and when they disappear 30,000 years ago, those stone tools look pretty much identical to each other. All technology across the whole range of Neanderthals is also pretty similar. When modern humans come, all that changes. Technology starts changing rapidly and becomes regionalized in different regions. Modern humans make different tools. And we have, after all, existed only a third of the time that Neanderthals have existed on the planet. And I think we agree that our technology today is quite different as 50,000 years ago. There are other things such as figurative art. Art that really depicts things comes only with modern humans. And of course, this thing that we spread across the whole globe. These earlier forms of humans lived for 2 million years in Africa, Europe, and Asia. Never crossed water, there you don't see land on the other side. Then come modern humans, and in 50,000 years, one colonized not only Americas, Australia, but every little speck of land in the Pacific. So the hope would, of course, be that some of the sort of reason for this very special thing that happened in human history would be hidden in this catalog of genetic changes that characterize present-day humans. So if we now, just to illustrate this, take a very strict definition and say, what are the changes that happen here that to our best of our knowledge is in 100% of all humans today when we look at all databases we have access to, yet Neanderthals and Denisovans look like the apes. Then that catalog is not a very big catalog of things. It's 31,000 substitutional differences, some insertions and deletions, and so on. So you can scroll through them in the computer in a few hours. So it's, for example, only around 3,000 regulatory regions. Now it's, of course, how you define regulatory regions, but if you take one definition of them, it's something like 3,000 changes. It's 96 amino acid changes. And I just focus on them for a second to sort of illustrate how you can go after this. This is not to say that regulatory changes are not perhaps even more important. So these 96 are actually located in just 87 proteins because some have several changes. And you can, of course, now start saying, could some of these be important? Hopefully there will be many experts on these genes that will sort of find interesting things here. We are, of course, biased to things that's saying things in the brain might be particularly interested. So we were quite interested when the Allen Brain Institute actually looked in different parts of the developing human brain and looked at these genes that have amino acid differences, how many of them are expressed there, and as a control, genes with silent changes that don't change amino acids, but otherwise are ascertained in exactly the same way, fixed in humans, ancestral in Neanderthals, and so on, and found that only in the proliferative zone, where the stem cells divide to make new neurons, do we see significantly more of these amino acid proteins that have changed amino acids expressed. And these are, of course, signals that rely on few proteins. It actually relies on 5 proteins, that signal. And quite strikingly, 3 of those 5 proteins are involved in the kinetochore and in the spindle. That surprised me a lot, but there are, of course, indications that say the mitotic cleavage planes of the stem cells in this epithelium when neurons are born determine, say, how many cell divisions you have and what types of neurons you make. So this is just sort of idle speculation, but perhaps these three proteins should be particularly interested in studies in brain development, what these changes meant. But that brings us to the last question then I had, and that is this question: how should we go after these types of changes that are essentially fixed in humans, yet are perhaps involved in human-specific traits, where we per definition have no sort of animal model? And I've gone around for years making jokes in the talks like this and saying what we want to do is take the Neanderthal allele and put in a transgenic human, and the human allele in a transgenic chimp, and said that we can't do that, of course, so we have to think about something else. But now it's suddenly not so ridiculous anymore, because there is people like George Church, a professor at Harvard, no less, that go around and it's even more extreme like this and say we should clone Neanderthals, we should put in all these changes in our catalog in a stem cell and make an individual. And I don't know why we are sort of forced to discuss this. I think it's technically impossible and ethically unthinkable, of course. But it doesn't sort of solve the problem, of course. We sort of, how will we go after these things? And I think that one thing that will happen soon, but people here are much better positioned than me to think and discuss this, is to say that I think in the future we'll be able to find back mutations in humans. When millions of people will have their genome sequenced when they go into the doctor's office, we of course know that all mutations compatible with human life exist in the human population because the genome is small enough and there are enough people there. But I think it will probably become ethically possible to find ways to go out and sequence and study these individuals. But that's a bit away. You can engineer these into iPS cells, of course, and we and others are in the process of doing that and study differentiation of cells in vitro. And what I want to make a little bit of propaganda for the last 3-4 minutes is then that one could perhaps after all use model organisms such as a mouse. And I want to illustrate that with one gene that we have studied since 9 years now. It's a gene called FOXP2 on chromosome 7 here. And it caught our interest because it encodes a transcription factor that when one copy is mutated, it sort of leads to severe language and speech problem in human families. It's also a very conserved protein that's encoded. So if you look in the mouse, it has only one amino acid difference here all the way to the chimp. But interestingly, 2 amino acid differences on the human lineage there. They are close to each other in one exon. So it seemed very interesting, first of all, to look in the Neanderthal genome and Denisovan genome to see if these changes are shared with them. And indeed, it turns out it is. So these changes are carried also by Neanderthals and Denisovans. So they happen back here. So they are not a modern human-specific one, but they are still interesting, of course, if they have something to do with speech and language, a human-specific trait. So what we have done already some years ago is to engineer these 2 changes into the chimp FOXP2 gene. So since this is such a conserved protein, we now have a mouse that essentially makes the human protein from its endogenous FOXP2 gene with one other very conserved change over there. So you then have this mouse, and you have the problem that you want to study a human-specific trait, speech and language. So we tried to speak to the mouse, but it didn't lead to anywhere. So instead we said this is a very conserved protein, so it could be anything in the phenotype really. So we were very lucky to work together with this mouse clinic in southern Germany that do a very comprehensive screen of then the homozygous knock-in mice with their litter mates that are wild type. So do look at very many phenotypes as to give you a feeling for it, to sort of going through them here. Hematology, what have you, eye function. All in all, they looked at 323 phenotypic traits. Not at all independent of each other. Many are, of course, highly correlated, but it's still a very comprehensive phenotypic screen. We had 2 independent knock-in lines we made, and there are only 2 phenotypes that were significantly different between knock-ins and litter mates. The first one is that the humanized mice are slightly more cautious in a new environment. measured so that you have a social group here, you open doors, and the mice can go in and explore an open area. Mice feel more safe along the walls, so the humanized mice stay more at the walls, whereas the other ones do mostly that, but also venture in the middle here. But it's just a transitory phase. So it's just the first few minutes are slower to explore the middle of the open field than the wild-type mice. I have no clue what this means whatsoever. The other phenotype, though, aroused my interest, and that is that they vocalize differently. There's no way to say they vocalize in a more human-like way, but you sort of measure that by taking 2-week-old pups out of their nest and a peep in the ultrasound area here, and the mom comes and brings them back into the nest. And there are a number of features in these sonograms where the humanized differ from the wild type. Sometimes it looks like sort of a dominant trait, sometimes co-dominant. So this is very interesting. It sort of supports that these changes might have something to do with muscle control in the oropharynx or something like that. But the frustrating thing is that there is sort of hardly anything known about the neurobiology of mouse vocalization, of course. But you, after all, now have an animal model. So you can take out neuronal precursor cells, for example. You can differentiate them in vitro. And you will find here that if you do that from the striatum, they actually grow out longer neurites. So things that will become dendrites and axons. And in the live animals, you can also see bigger dendritic trees in the striatum. You can do electrophysiology in slices from these things, and you can see that long-term depression, where sort of the neuronal excitability goes down after a high-frequency stimulation, a sort of model for learning and memory, is much stronger in the striatum in the humanized mice than in the non-humanized mice. So what we then know about this, it alters vocalization, you have more dendrites, you have this increased synaptic plasticity, and you have decreased dopamine levels that I haven't mentioned. So it sort of generates a hypothesis. A reasonable hypothesis is that we would have something to do with corticobasal ganglia circuits, where the basal ganglia receives input and gives output to the cortex and the brainstem that has to do with motor learning often. And there are other sort of things that support this, the regions in the brain where we see these bigger dendritic trees. Just very recently then, a previous graduate student, I worked with Anne Graybill at MIT and looking at motor learning in these humanized mice. So they do different tests where the mouse have to sort of learn to go towards a signal to get some reward. That can be a tactile signal here or a visual signal. And under certain paradigms, these mice learn in 7 to 8 days what the wild-type mice will take 11, 12 days to learn. And by doing various variations of this is not my field, so I just relate what they have found. They have sort of made it likely that this has to do with a switch from a sort of what they call a declarative phase of learning, where the animal needs the cue, knows I goes towards the lamp, and then after a while you can stop turning on the lamp and it will know it will go to the left. It will sort of come to a procedural phase where it sort of endogenously know I just turn left to get my reward. So they illustrate this by saying if you think about how you learn to bicycle, in the beginning you think about how you bicycle and you're very bad at it. And then after a while you sort of automated it and you get very good at bicycling. And this correlates with an activity shift from the medial part of the striatum to the lateral part of the striatum. So this generated a sort of hypothesis that we would see some difference in synaptic plasticity medially and laterally. So we then went back and did the electrophysiology in the mice, and in the lateral part of the striatum we see this increased synaptic plasticity, increased LTD, whereas when we then look medially, we see no effect at all, or if anything, an effect in the opposite direction, but no significant such effect. So this then seems to sort of support the current hypothesis that these amino acid changes have something to do with changing corticobasal ganglia circuits to make for faster proceduralization, sort of automation of learning, and perhaps then with aspects of learning speech and language. And you can, of course, make a nice sort of speculation and say the sort of what we learn to do when we learn to speak is that we automate coordination of muscle movements in oropharynx to produce articulate speech. And it's probably the most sophisticated muscle coordination that we actually do in our life. And a form of thing that, say, apes are not able to do. I should accelerate a little bit here. I should just say, in the FOXP2 gene, we also find a regulatory change, it seems, that is very conserved all the way to frogs, yet changed in modern humans. And it affects the PAU3F2 site, where we can show that the ancestral version actually binds less of the dimer and also is more efficient in driving expression in the model system. But we don't know anything more about that, but it somehow seems that we have these amino acid changes and perhaps a regulatory change then there. So this is a lot of work over several years, of course, and in the end, we sort of have to study these changes, I think, in this systematic way back into the organisms, to then hopefully maybe in the end understand something of what set humans on such a very special track among all other primates. So I hope I've sort of convinced you that if you're interested then in human evolution and recent changes in humans, it's very good to have the genomes of our closest relatives, because you can sort of see what have happened on this line here, in the future also here when we have more genomes here. But it will not be enough to have the genomes. The genomes are just a tool, of course. What we have to do in the end is sort of functional work. And I think the most reasonable way forward there is, after all, a humanized mouse. So there are many people involved in this, many more than I can mention. I'd already mentioned that in the analysis consortium, Jim Mulliken and his group here played an important role. I also want to mention just one person, Matthias Meyer in the group, that came up with this single-stranded library protocol that really made it possible to generate these high-coverage genomes from these extinct forms of humans. And with that, I thank you for your attention.
We have microphones in each aisle, and so if people have questions, I'd ask them to please go to a microphone. Yes?
You mentioned at the beginning that mitochondrial DNA did not support a mixture hypothesis. And later you said that, wouldn't this imply that the DNA from the Neanderthal was male?
Yes, so that would be one explanation for it. It's also compatible with that this is just stochastic variation. The mitochondrial genome is inherited as just one unit, so it's rather easily lost if a female has no female offspring. It's sort of her lineage is lost. So the more boring explanation is that. Now, we also see on the X chromosome reduced contribution from Neanderthals. When we first saw that, I said, that's clear. It has to be male gene flow because, you know, males transmit only to half their children. They transmit an X chromosome. The other ones get a Y. But then we've seen these sort of deserts in other chromosomes too. So that has made us shy away from saying that this is male gene flow. But if I would guess, I would say it's mainly males.
Okay, and doesn't this conflict with what you said later on, that Neanderthals had probably less fertility?
Well, the hybrids would have had less fertility. Okay. So I think in the hybrids, this would suggest that the female hybrids may have been fertile, but the male hybrids may have reduced fertility.
Thank you.
In the analysis that led to the testes-specific gene standing out, in speciation and model systems, heterochromatic regions can be very important. So do you see any signals pointing you towards those non-genic regions?
And the only analysis we have done is that in these deserts what genes are enriched there. So there might well be, we would not have seen it as we have analyzed it. Much of the heterochromatic things are perhaps also repeated, or so there we have really a mapping problem from our short little pieces. So we can only statistically say how many copies of a repeat is there. We cannot really reconstruct the array of repeats.
So Svante, it seems that you're trying to identify humans with back mutations, as you called them, is not a crazy idea. I mean, it's absolutely going to be probably feasible in only a matter of years if we can get all the data aggregated in a way you can look specifically. But how are you imagining then taking that knowledge to sort of the next study? I mean, if you had a person in front of you with some of these, I mean, are you going to study the individual? You're going to study their cells? You are imagining just immediately going to mice? But it would seem to me you'd want to have the opportunity of taking advantage of having a person.
No, I would, after all, hope that one would find this in cohorts with a recall possibility.
And what would you do with those individuals?
Well, that, of course, depends on what gene or what regulatory element is affected, where you would
So
look
you'd want to
at the
go into
phenotype.
that with some functional insights
Yes,
already done experimentally
I would
through mice or
imagine
other
that, yes. And before we get to that, I would propagate that on genotyping arrays, these are just 31,000 SNPs. Why don't we add them to genotyping arrays today so we can pick them up quicker, actually?
I'm a little troubled to understand that your statement that Neanderthals didn't exist in Africa at all. If they came out of Africa 300,000 years before, none of them stayed, or they only developed, or they radiated, they couldn't walk back to Africa.
So it's my sloppy language, I guess. It's the ancestors of Neanderthals that come out of Africa. So there are things that paleontologists would call Homo heidelbergensis, that you see both in Africa and outside Africa. And the idea is that they evolve into Neanderthals. But the morphology we use to define Neanderthals is never seen. That's something that appears in Western Eurasia. And we now have a little bit of DNA sequence from Homo heidelbergensis that's 400,000 years old. So we begin to be able to reconstruct this, perhaps.
The DNA evidence is, I can see that link being there, but the fact we haven't found a Neanderthal in Africa, it could be found tomorrow.
Yes, but I would still be surprised if one did that, because, yes, there has been, after all, quite a lot of things found also in Africa by now.
Okay, well, we have one more question. Last question.
Just quickly, the FOXP2 story is so compelling. Are there any other kind of specific gene function stories that are evolving in your lab or elsewhere?
Well, so we are now trying to put in a few others of these more recent changes into mice, 3 of them that we sort of bet on will have some phenotype. There are other studies in mice that have come along. There has been this EDAR gene that is to do with sweat glands and hair structure. That's a variant among humans today that have been put into mice and give a sort of reasonable expected phenotype and even expectation of phenotypes that you then could go out and check in the human population and find them. So I think it's not unreasonable to suggest that mice can be, for some things, a good model, but it has, of course, its limitations.
Okay, well, please join me in thanking Svante for just a terrific presentation.